treatment, we both treated the catalyst as well as a reductant, as well as a protonating agent by a DFT. And here you see the energy profile of this process. So here we have the bare molybdenum complex. It first binds N2, and this is the most endergonic step. Here, the first protonation of N2, and then we have your reduction and so on. Here you have the NN cleavage, giving rise to the first molecule of ammonia. Here you have the nitrido complex, and here at the end you have the amine complex, and here you come back to the dinitrogen complex. So there are maybe three important things with this cycle. First of all, this or the most difficult reaction is the first protonation, and this NN cleavage is actually very exergonic, and this process is strictly alternating in protonation and reduction. And we got some criticism because we started with the small models, and 10 years later, we basically made the same calculation with a full model of the catalyst. So these calculations took over 1 year, but basically we got the same energy profile, which is just due to the fact that these residues here are perpendicular to the lone pairs of these amido nitrogens. So this was already quite a good approximation. And if one compares the theory with the experiment, so here you have all the protonation and reduction steps. Here you have the experimental values, and for certain reductions and for certain protonations, we have experimental values, and you can see that the reductions are very well matched by the calculations. So these are our calculations. So, for example, for the reduction of the NNH2 complex, we get -5 kcals, and experimentally it says -4 to -7. Or for the reduction of the imido complex, we get -7, and experimentally it's -5 to -6 kcals. Or here we are also very close. This is a reduction of the amine complex, and also for the exchange of the N2 by ammonia. The ammonia by N2, we get a very good value if we can compare this to the experiment. What is still not good is the calculated values of the protonation. So consistently we get 10 kcals, too exergonic for these steps, which is probably connected to the fact that these protonations are not properly modeled by us yet. Okay, now we come to the chat cycle, and here you see a picture of the chat cycle. We start with a bis dinitrogen complex. We basically go through the same intermediates as in the Schrock cycle. So you also have a diazonido minus, a hydrozido 2 minus, a nitrido complex, imido, and so on. But from the established chemistry of this chat cycle, the steps are less clear-cut than in the Schrock cycle. So it's not alternating. Here you have 2 protonations, and then you have a 2-electron reduction, and so on. And of course, this system has never been catalytic in the past. And we did a lot of model chemistry to understand this cycle, and it appeared to us that there are 2 basic problems. First of all, we have this dissociation or exchange of the transaxial ligand. So we are here at the molybdenum 1 stage at the amine complex. We have to cleave off this X ligand to make the bis dinitrogen complex, reduce it, and then in the first step, one of the nitrogens is cleaved off again, and here we have again the X ligand. So this is a barrier to a catalytic reaction, and more importantly, at this molybdenum 1 stage, we have here disproportionation. So we have here a molybdenum 1 amine complex or dinitrogen complex, and it disproportionates into a molybdenum 2 complex and the molybdenum 0 complex. And this is basically lost because it cannot be reduced anymore. So what we decided to do was to include this transaxial ligand in a polydentate ligand system. And we started with two types of systems. Here you see these tripod systems with tripodal ligands, so a combination of a tripodal ligand and a diphos ligand. And here we looked at systems with tridentate ligands. And here you see the central donor, which we took as a phosphorus, nitrogen, or carbon. And here we varied this donor atom, and here we varied the substituents. So as I said, we used here DPPM or DMPM co-ligands, and we investigated the influence on the activation of N2 and the properties of the complex. And after this phase, we decided to combine these two approaches. So we combined the tripod and the trident system into this pentapod ligand. This is a pentadentate tetrapodal ligand, pentapod ligands. And in that case, this transphosphine, this ligand X, should be strongly tethered to the molybdenum center. Yeah, here you see the synthesis of this pentapod ligand. So we basically started here with this diallyl phosphinic acid ester, which we phosphonated in a radical reaction, and reduction leads to this propyl PPHP ligand, which we also used as a tridentate ligand to make a dinitrogen complex. Here you see this dinitrogen complex. The N2 ligand is moderately activated. So this showed us that this tridentate ligand is here a suitable building block for pentapod ligands. For this pentapod system, we also needed a tripod component. We made this as shown here. So by selective phosphonation of this trichloro precursor, and we coupled this to the propyl PPHP in the presence of Bulli, and arrived at this first version of this pentapod ligand, the P2P2Phi, which was then coordinated to molybdenum 3 and subjected. This molybdenum 3 precursor was subjected to an amalgam reduction, giving a dinitrogen complex. But unfortunately, this was not a mononuclear complex, but as we believe, is a polymer. So we needed a new approach to make such a pentapod ligand. And we started from another system. Here we took this dichloropivaleic acid, reduced it to the alcohol, converted it to the triflate, and coupled this with a propyl PPHP, which led to this asymmetric ligand, which has now 3 alkyl phosphine donors and 2 diphenyl phosphine donors. And we also bound this to MOCl3 THF3, so the trichloro tris THF molybdenum 3 complex, and ended up here with this molybdenum 3 system. And we believe this is an axially coordinated complex because it has the same EPR spectrum as this well-defined tripod system here. And if we subject that to an amalgam reduction, we end up here with the pentapod complex. And last year, we discovered that this complex acts as a catalyst for the reduction of N2 to ammonia using samarium iodide with water. We achieved 26 equivalents of ammonia. And for this, we also got a cover picture of Chemistry European Journal. This appeared last year. So what is what is special on this complex? And to this end, we compared it with two other complexes which we have made before, which do not have that pentapod coordination. So this complex you know already. It has this tridentate ligand, and here this has a standard tripod with the MPM. And the most significant feature is that this complex here has the most activated ligand. So this is this NN stretch is very low, and as a matter of fact, it has the lowest NN stretching frequency of all molybdenum 0 complexes. So certainly one important point is the strong activation of this N2 ligand in this pentapod system. But we also have structural features which makes this complex special. Here you see the NN distance, which is not much elongated relative to free N2. And here you see the axial, the bond length to this axial phosphine. And quite remarkably, this axial bond length is very short, and it's much shorter than in the other systems. So in 1 and 2, we have longer axial molybdenum phosphine distances. And in order to learn more about this molybdenum P bonding, we also studied the NMR spectra. Here you see the 15N NMR. Here you have you see the resonance of the beta nitrogen, which is split into a doublet by coupling with the alpha nitrogen. Here you see the signal of the alpha nitrogen, which is split by coupling with the beta nitrogen and the other phosphines. And also this 31P NMR gives us important information about this bonding. Here you see the 31P NMR of the N14 complex. It gives rise to a so-called AA prime XX prime M spectrum. So here you have the XX prime nuclei, here you have the AA prime nuclei, and here you have the M nucleus. And in an AA prime XX prime spectra, you have 10 lines in each of the subspectra, and by coupling with this M signal, they double. Each signal doubles, and you get 20 lines here and 20 lines here. And we saw that when we made the N15 complex. Here you have the experimental spectrum of the N15 complex. Here you have a simulated spectrum. And it was immediately clear that the experimental spectrum has more peaks than the simulated spectrum. And it turned out, and we now go to this M spectrum, and it turned out that this 15N complex also contained some amount of the 14N complex. So you have here you see the spectrum of the 14N complex in black, and here you see the spectrum of the a mixture of the 14N and the 15N spectrum. Here you see the 15N spectrum, and remarkably, every peak is split by a coupling of an alpha with this phosphorus, and here you see the additional spectrum by coupling of this phosphorus with N-beta. And remarkably, this 15N spectrum is shifted with regard to the 14N spectrum. So what we have is a second order isotope shift of the 31P M signal upon substitution of 14N by 15N, and this is due to the unharmonicity of the potential in this molybdenum N bond. So if you take a heavier ligand, which is 15N, it is a bit closer to the molybdenum center, and it makes a different interaction with the transphosphine. It's very remarkable. Now we go to the NNH2 system. We can protonate this N2 system here with H-barf in dichloromethane, and this protonation is possible under retention of the pentapod coordination, which you see here. You have the AA prime XX prime M of the dinitrogen complex, and here you have this of the NNH2 complex, and you'll see the AA prime shifts and the XX prime shifts, and the M shifts much more than the other two signals, which is due to the fact that you have a triple bond here, and due to the trans effect, this bond here is elongated. And we also studied the 31P NMR spectra of this complex, and again I go to the M spectrum, and again we have this mixture of the 14N and the 15N. So in black you see the 14N, and here you see the 15N, and now we see that it's all symmetric, so we don't have this isotope shift upon the of the 31P M signal upon substitution of 14N by 15N, which is due to the fact that this molybdenum N triple bond is much stronger than the molybdenum N bond in the dinitrogen complex, and correspondingly the anharmonicity in this bond is much less pronounced than in the dinitrogen complex. Okay, here you have the proof that we have an NNH2 complex. So here we have the 14N signal, here the 15N signal, the 15N this is the 1H15N HMBC. So the proton signal is split by coupling with the N beta, and the coupling constant is 94.6 Hertz. And I also go to the N beta signal, and here you see the splitting due to the coupling with the protons, and then we have an additional splitting due to the coupling of the N beta with the phosphine, and of course also with the N alpha. Importantly, this NNH2 complex also served as a catalyst for the reduction of N2 to ammonia. So we could equally well use this NNH2 complex as a catalyst in the presence of samarium iodide and water. So it converted N2 to ammonia with very similar turnover number. So we think that this proves that this mechanism follows a mononuclear pathway. So we go from the N2 complex over the diazonidal to this hydrazidal 2- complex, and further on here. And actually it was a question whether these systems follow a dinuclear system, a dinuclear pathway based on a publication which came out in 2019. So Masuda had this complex, it's a chat type complex, if you oxidize it to the molybdenum 1 complex, it makes a dinuclear complex and it splits the N2 to the nitrido. And we are convinced that this is not the pathway in our system, because also our system cannot form a dimer. So I'm at the end of my talk. I want to summarize. I first showed you the Schrock cycle. Then we went to the Chat cycle. We identified mechanistic problems. We made this pentapod ligand, the derived complex, serves as a catalyst for the generation of ammonia from N2. And we believe it follows a mononuclear pathway. And this would mean it's a first phosphine-based single-site molybdenum catalyst. Now, of course, we want to generate further intermediates. We want to optimize the catalytic activity, and we also want to optimize or modify our pentapod system, both with respect to the metal centers and with respect to the composition of this ligand. I have to thank my students, the spectroscopy people in my department, and also Castianeta for X-ray studies. And of course, I also have to thank you for your kind attention. Thank you.
Thank you very much, Professor Chisek, for this excellent presentation. Are there any questions?
I have a question.
If there is no question, I have one very quickly, if I can say this. You know, the unsymmetrical nature of the metal nitrogen bond that shows up in the spectra, that is remarkable. The anisotropy is very, very small, and it still is picked up.
You are speaking of the structure.
Yes, before and after adding the hydrogens. Yes, exactly. The molybdenum nitrogen bond is not symmetric, but how asymmetric is it in terms of any other parameter other than the split of the spectra? Or what is the measure of it in any other fashion?
Pardon? I did not quite get your question. What do you mean by symmetric and asymmetric?
No, the molybdenum nitrogen bond.
Yeah, in the N2 complex.
Yes, is not symmetrical. That's what you mentioned, right?
Oh no, you have an anharmonicity in the potential, which yes, means which means if you increase the mass of the N2 ligand, it goes closer to the metal. So if you go to the essence of anharmonicity is if you go to higher vibrational levels, you go the metal ligand bond distance increases.
Yeah. What is the magnitude of this change? That is what I wanted to know. Roughly, what would be the magnitude of this change?
You mean whether I can tell in terms of angstroms how much closer the N15 is to the molybdenum than the N14?
Yes.
I cannot tell. I would have to calculate this. I think it's very little. So first of all, I've thought about how to do this. You first have to derive or evaluate this potential. You have to fit it to an anharmonic potential, and then you have to determine the metal ligand bond distance in the different vibrational levels for the different isotopomers of the N2, and we have not done that yet.
Yeah,
but it would be interesting to do.
Yes.
Yeah, I agree.
It's so small.
Yeah.
Yeah, that's right.
It's probably less fraction of an angstrom. Yeah, it's very little. But this is obviously a very sensitive measure of this metal ligand bonding.
Yes, we have another question, Professor Narasimhamurthy.
Yeah, I have a question. Actually, the natural enzyme nitrogenase uses multi-center molybdenum and iron. And where do you think in your case molybdenum will it be able to provide all the electrons to reduce nitrogen to ammonia?
So nowadays it's clear, all scenarios agree in that the N2 is not bound to the molybdenum. Is not bound to the molybdenum. It's bound to the iron, but the special coordination mode to this iron centers is not clear yet. It may actually displace one of the bridging sulfur atoms. So that's a new hypothesis, and but this question is still very open. But the molybdenum is not involved in the bonding. On the other hand, I mean, the reason why I have started with molybdenum was that for molybdenum, most of the intermediates have been available. So all the scenario has been set up, but the system was not catalytic. So I did not choose molybdenum because it was contained in the female core, but because it has the potential of a catalytic action. But there are many other differences of these systems to nitrogenase. Nitrogenase is a cluster, and these are mononuclear systems, for example. So it has not. I'm afraid to say that, but I believe what this model chemistry on mononuclear molybdenum centers has not much relevance to the iron molybdenum cofactor.
Thank you, Professor Felix Tucek, for this excellent presentation. I would move on now to the next speaker, Professor S. Ramakrishnan. Professor Ramakrishnan does not require an introduction, and so I do not give an introduction. So, Professor Ramakrishnan, please upload your presentation and begin. Strategies for reducing length scales of microphase separation and more. Professor Ramakrishnan.
So, is the slide visible?
Yes, go ahead.
Okay, and my cursor as well. So yes. Firstly, thank you very much. Thank you, Tilagar and Debashish, and of course, Arunan, for asking me to give a talk. It's really a pleasure to give a talk at this symposium. So this is the title, and so let me move on quickly without wasting too much time. So the idea is that, this is something that you might have seen before, many of the students. If you mix two polymers, let's say, they typically don't mix, and the size of the domains formed are not thermodynamically governed typically. It depends on how you mix it. In some sense, it's like paraffin wax melted and stirred in water. And if you stir it very rapidly and cool, you'll get finer droplets of wax in water. And if you don't stir it so effectively, you'll get larger ones. So it's largely a process-dependent size. So people have been thinking about how to control the size. And one way to do it is to tie the two polymers at one point. Okay. And so these are called diblock polymers. And when you tie them together, the blue and the red block has no choice but to remain in the vicinity of the other. And when you do this, when you impose this constraint, what happens is the dimension of the domains that are formed, or the microphase separated domains that are formed, depends now on the size of the red block and the size of the blue block. Okay, so in other words, one will be able to control the size of the domains formed in this kind of system by simply changing the length of either the blue or the red block. Okay, so that's this basic idea. Now, this has been well understood in blockopolymers. So if you change the relative size of the red and blue block, you can go from spheres all the way to cylinders, gyroid, bicontinuous structures. Finally, lamellae, when they are equal volumes, you get to lamellae, and of course the inverse structure. And this is the theoretical phase diagram that was published many years ago, over 25 years ago, which actually tells you the various kinds of phases that are formed in a diblock copolymer. One of the important parameters that govern the nature of this phase separation or this curve of the is the segregation strength, which is simply chi and n, where chi is the Flory-Huggins interaction parameter and n is the size of the macromolecule. Okay, so and chi is intrinsically dependent on the fundamental or the structural features of the blue and red block. In other words, the chemical composition of the blue and red. And you can't increase the chi very much. What is important to remember is in this phase diagram, unless the chi attains a certain value like 11.5, below 11.5, it's actually not microphase separated. So it has to exceed 11.5 to see all these beautiful phases that are there. Now, since chi cannot be increased enormously, it puts a lower limit on the value of n, so that chi n is actually 11.5. And once you have a lower limit on the value of n, the size becomes limited because the size depends on n. Okay, and therefore, typically in block copolymers, it's very hard to get dimensions that are smaller than 10 nanometers. Okay, that's the limiting factor if you use diblock. Why are we interested in diblock copolymers with very small dimensions? There are many reasons. I just picked up two of them. One is block copolymer lithography. Lithography is the process by which you can transfer patterns which are extensively used in, say, generation of silicon chips. But usually it is done top down. You use a mask and you irradiate and remove material where the light is not shown. But here people are trying to develop self-assembled mechanisms for microphase separation. So that's one way. The second interesting and important application is for nanofiltration. So I told you can form cylinders of one in a matrix of the other. And if you can get the cylinders to stand vertically, which you can, then you can remove one of the portions. If you remove the cylinders, you actually have holes. The dimension of the holes are actually governed by the dimension of one of the blocks. Okay, so these are some interesting applications. People have been interested in this. They have used a variety of techniques. Johnson at MIT has taken a diblock copolymer, and at this junction, he has a polymerizable unit. So he stitches the junction, and so this is the polymer backbone, and you have these red and blue pendant units on either side of the backbone, very closely packed. And when they are very closely packed, the lamellae that are formed when the dimensions of these two are roughly similar looks like this, where the backbone itself is actually parallel to the interface between the two lamellae. Okay, so the backbone lies like this, and the side chains, two side chains, protrude into the two alternate domains. So he was able to show that you can control the dimension by changing the size of the red and blue block. Likewise, Mahantappa in Wisconsin also did some very interesting work where he took a triblock copolymer, ABA triblock, and right at the center of the triblock, he had a polymerizable unit and stitched it up. Both of these are strategies that use what are called brush polymers to generate microphase separated morphologies. And the important thing is they were able to get dimensions which are very, very small. Okay, so these are the two strategies sort of roughly drawn. This is Johnson and this is Mahantappa. And what we have done in the last almost a decade now is to explore an alternate strategy where you have these blue segments that are immiscible with red segments periodically placed along a backbone. Periodicity is important. That means the distance between these two, the contour length of the red segments between two adjacent blue segments are exactly the same. Okay, and what we will show you is very quickly, because I want to take you through this quickly, is that when the length of the segment is large enough and it strongly dislikes the blue segment, then one way that this chain can avoid contact between the red and the blue segments is to fold in a zigzag fashion, so that it collocates all the red segments at the center and throws out the blue segments on either side of this chain. So this is an alternate strategy. And in this strategy, what we are able to show is that the dimension of phase separation simply does not depend on the length of the polymer, but only depends on the length of this red blue segment and the length of the red segment, as I'll show you. Okay, so this was the first effort, one of the early efforts, not the first. We did earlier work, Raju did some work, but I just want to illustrate this idea that Sananda did. she took this itaconic acid, which is a very cheap bio-sourced diacid, and you simply link it, polymerize it with a diol to make a polyester. So this is a polyester. And what is nice about this polyester is that it has a double bond conjugated to a carbonyl, and therefore it becomes a nice site for a Michael addition. In other words, you can make a very hydrophobic backbone. Here is an example of a C20 chain that lies between two adjacent double bonds. Okay, and so now you can do a thiol-Michael reaction, which means you can take a polyethylene glycol terminated with SH on one side and OCH3 on the other, and park it on every location here. And so what she did was she changed the length of the blue segment, which is the polyethylene glycol segment, which doesn't like the hydrocarbon segment. And so this segment here is fixed while the length of the blue segment is increased. Okay, so you go from all the way triethylene glycol to all the way PEG 750. So these numbers here reflect the length of the PEG segment. So we increase the PEG. So what you see in a DSC is quite remarkable. One thing you notice is one of the phase transitions, one of the melting transitions in the DSC remains roughly constant. And remember, I told you that the segment that remains constant is the hydrocarbon segment. So we assign this first melting peak to the melting of the hydrocarbon. And when the PEG segment increases in length, it also begins to crystallize. And you see a second melting point, which corresponds to the melting point of the polyethylene glycol. Clearly, this was one evidence that the zigzag folding and co-location of the hydrocarbon and PEG actually happen, so that independent crystallization of both the backbone and the side chain can happen. Okay, so now you can do the small angle X-ray scattering, and then you see these lamellar faces that are formed, and you can measure the lamellar dimension by looking at the scattering pattern. And what you see is the lamellar dimension increases as you increase the segment of the blue. Okay, and you can plot the interlamellar spacing as a function of number of atoms in the PEG. In some sense, it is the volume of the PEG, and you see as the volume of the PEG increases, the interlamellar spacing also increases linearly. Okay, so that's essentially dependent on that. So what Ramakrishna did was, he said, instead of changing the length of the pendant segment, you can also change the periodicity at which these segments of fixed length occur. And so he developed an alternate strategy where the red segment, that is a zigzag folded segment, actually increases. And he was also able to show that the interlamellar spacing actually varies linearly with the length of this. Okay, so in other words, the backbone actually undergoes a zigzag folding. This is a matter of detail, I'll skip it. So in some sense, the summary really is that you can change the periodicity of the occurrence of these segments. That means this, or you can change the length of this, and both of which allow you to control the segment. Now, Gabriel went on to do something else. So what we did at that stage was to ask the question, suppose you put in segments that like to interact with each other, either on the pendant or on the backbone. Can these segments actually also enable these systems to fold and also give some interesting liquid crystalline properties to these systems? I won't go into the details here, but just to summarize, a very similar behavior as we saw in the earlier case, we can see in these systems as well, that the interlamellar spacing depends not only on the segment, the brown segment, but also on the green segment in both these cases. But here, these two are examples where in one case, the mesogen is present in the pendant unit, and in the other case, the mesogen is present in the backbone. And this pendant unit is something, we call it a folding element. That means it drives the polymer simply to fold, but then the mesogens actually layer. So in these X-ray scattering pit profiles, what you see is the distance between the adjacent mesogen layers. What we did, even more interesting, what Gabriel did was, he now then included two different kinds of mesogens, one in the backbone and a different one in the pendant unit. And when you do this, the lamellae forms in such a way where the interlamellar spacing now is actually the distance between the, let's say this is an azobenzene and this is a biphenyl. So here is a very interesting way to layer functional segments at very precise distances in a layered organization, okay, and two different functional entities. So this can be one entity and this can be other entity, and the distance between this in the layered ordering is governed primarily by these two segments, okay, which is tunable from a synthetic point of view. So really, we can now begin to think of additional functions that you can impart to this folded systems. So yeah, this is the summary of what I said. So before I sort of move on to something, what I said and more, I see that I have 13 minutes left. What Ramakrishna did was, I told you that if I have periodically located segments of one type on a backbone, it folds in a zigzag fashion to put these brown segments on either side of the folded chain. Ramakrishna developed a strategy where you can put two different segments, but located alternately. Okay, and so you can put a brown segment in every alternate location and a green segment in every other alternate location. And if brown, blue, and green, all of them don't like each other, it zigzag folds. But now you have developed an asymmetrical system or unsymmetrical system where you have one functional entity on the top and another one on the bottom. And now what he's gone on to do, which we are now working on, is that he's put a chromophore on one end with a hydrophilic segment, but only a hydrophilic segment on the other. So if it folds, it causes all the chromophores to orient in the same direction in solution. Okay, and so we are now examining this property of these things using second harmonic light scattering in collaboration with P.K. Das and his group to find out if we can probe the way the polymer folds if you have chromophores that begin to orient in the same direction as the folding happens. Okay, so now I'm going to go to the and more part. So that was just a quick summary. So there were three different talks, and this is primarily cheers to the numerous lunch walks that I have with Professor Samuelson every day, and for his patient listening and probing and being a probing, gentle critique of the ideas that I have. So instead of talking about all the successful experiments that we've had, I want to talk about some experiments that we didn't succeed, but I think they're interesting experiments that might actually provide some valuable information. So I'm going to talk quickly about three different experiments. One is simultaneous probing of crosslink density and rheological properties of polymer networks. That's the last one. Photo-induced generation of a conjugated polymer. And the last one is actually really a thought experiment of mechanically induced enzyme mimics. Okay, let me go quickly. There's only one slide on each, so I don't think I'll take too much time. So this is malachite green, and malachite green is a very old dye. We all know that. What is interesting about malachite green doesn't have this double bond, is that if you have this species, if you shine light on it, it does something very interesting. It dissociates into an iron pair. Okay, and if you just let it remain at room temperature, it gradually goes back into the covalent adduct. And as soon as you generate this cationic trityl unit, it becomes intensely colored. So in other words, this transformation should be easily monitorable by the generation of a very intense color. So the idea that we had is to exploit this sort of light-induced cleavage of a bond to generate two entities, an ion pair. Okay, so what we wanted to do, in fact, Gabriel started with this, but we didn't succeed, is to make a crosslinker. Just to tell you that when you have a molecule that has two polymerizable units, two double bonds, it's a crosslinker because when you polymerize this forms a part of one back chain, and this can form a part of another chain, and therefore the entire polymer becomes a crosslink network. So the idea is if you polymerize this or co-polymerize this with monomer, this would become a crosslink junction, okay, which is holding the network in place. And if you shine light, of course, the cross-linking will cleave and form an ion pair. And today you can do rheological measurements using a transparent disk, which means you can simultaneously measure the absorption spectra of the polymer while you're actually shearing the system. So if you can shine light while you simultaneously do this rheological measurements, you can actually change the extent, or you shine light and cleave it and watch it recover as a function of time while you monitor the UV spectra and the rheological properties simultaneously. You can get both a handle on the number of crosslinks that are present from the absorption spectra and the evolution of rheological properties as the network evolves. Okay, so that's one experiment we failed, but we hope maybe some other day we'll do it. Another experiment has the same sort of unit, trityl unit, And you will see here, that is, the idea is to photo trigger conjugation. So what exactly do I mean by this? So here is a trityl unit, triphenyl methane unit, and it has a halogen atom connected to it. And halogen atoms are known to homolytically cleave on shining light, especially when you have a very stable radical like a trityl radical that is formed. So the idea was to make a conjugated polymer or a non-conjugated polymer that looks like this. It has an sp3 carbon, so it's not conjugated, but it has a double bond here, right? So I've just rewritten this slightly differently. So this repeat unit is gone on this side for ease. I'll tell you why. So this renders it non-conjugated. You shine light, this homolytic cleavage happens to generate a radical and a radical in the backbone. And if you have a radical, this radical and this radical will actually combine to generate a conjugated backbone. In other words, you can now, if you design this polymer and you're successful in making it, you can now trigger the transformation in the solid state from something that is non-conjugated to a highly conjugated system. And there's a dramatic difference in properties, not only that the color becomes highly intense when you have long conjugation, but also transport properties of the system can vary dramatically. And since this is light induced, in principle, you can pattern this pre-polymer to draw circuits if you succeed. Okay, and the last idea is actually enzyme mimicking, what's a chemist's approach. Okay, so the idea is that now there is reasonable agreement that the turnover rates in an enzymatic reaction is very strongly controlled by conformational motions of the enzyme. Okay, that means and a recent paper, 15 years ago roughly, showed that NMR studies, the movement in the enzyme actually correlates very effectively with the turnover rate. Okay, so there is a nice correlation between the two. So from a chemist's point of view, people have been able to trying to mimic this by simply placing the active sites of the enzyme in a very controlled location, either in a macromolecule or in an imprinted matrix. And what they do is use a transition state analog of the reaction they want to mimic. Okay, and so they construct these binding units around a transition state analog, and then remove the transition state analog so that the positions of these binding units for the analytes is fixed by some mechanism. Okay, and the idea is then you come up with the two entities that are going to bind with this. Let's say this is a reactant A, and then there's a reactant B that comes and binds. And now, of course, they are positioned appropriately for the reaction to happen, and you generate a product, and the product should unbind. But the tough problem is that if you've designed the system so perfectly, the difference between the transition state analog and the product is very little, and therefore there's very little motivation for the product to leave once it is formed. And this is basically what is called product inhibition, which lowers the ability of the enzyme to function effectively. So how does one get over this? Okay, and the idea was the following. So the thought experiment, this is a thought experiment, no experiments have been done to try this. The idea is to take an enzyme substrate complex or an equivalent, a chemist's equivalent of it, and trap it in a polymer network. You can trap it covalently. In other words, some portions of the polymer chain are actually linked to this polymer network. And this network has an elastic network, which means it has a low glass transition temperature, so you can stretch on the network. Okay, and so the idea is if you stretch it out, and if you've done the experiment right, you would displace, you will sort of move these sites away and cause the effective binding constant of the product with the network to go down. Okay? And so it weakens the binding. When it weakens the binding, the substrate leaves, and then you de-stretch it, you contract it, the conformation recovers the original conformation, and then another two reactant molecules can come bind with it, and this process can go on. Okay, so in other words, if I have this cycle, I apply an oscillatory shear in this network, I simply stretch, and I can change in a machine like a dynamic mechanical analyzer. You can actually change the frequency at which you lose oscillation. And so the thought experiment is that as you change the oscillation frequency, you can actually map the arrival time of the substrates. In other words, the rate determining step in these cases many times is the arrival of the two reactants to the reactive site. So by varying the frequency of oscillation, you might be, and studying the rate of the chemical transformation as a function of the frequency of oscillation, one might be able to measure using a mechanical transformation some idea concerning the rate of these processes. Okay, so that's my last slide. And so I'd like to thank, before I close, the students who have done all the work that I presented in this meeting: Joydeep Mandal, Sananda Chanda, Ramakrishna Sarkar, and Gabriel Orodopo. And my collaborators on this have been Bojay Gaud and DST, DAE, and IISc for funding. And of course, I'll leave you with this slide, which, as I was trying to search for some photographs of Ashok, and I could only find this. And I think this is a friend of Ashok. I forgot his name, but there's a friend of Ashok who was visiting, and we took a photograph in my lab. Must have been about 20 years ago. And this was thanks to the chairman of this session. He gave me this photograph, which was probably taken about 40 years ago. I don't know how many of you can recognize the chairman and Professor Samuelson in this, but both of them are there. So good luck in recognizing them. So thank you. I hope I've stuck to time. Thank you, Jemis. I'm done with the presentation.
Thank you. Thank you, Professor Ramakrishnan. You did stop within the time. So we have time for questions. Well, I think, I see one hand raised. Please go ahead.
Binny has a question.
Binny, please.
Nice talk and very interesting work, RK. I was interested in some of the stuff that you were doing in the microphase separation experiments.
Okay.
And I was wondering whether you looked at any of the phase diagrams for those systems, and whether you had any thoughts about any limits, any constraints on the size of the Flory parameter for those systems.
Binny, in this, in graft copolymers, as well as in Johnson's and Mahantapa's work, the Flory parameter plays a little role, very little role, because the constraint comes largely from two things. In our system, I can tell you the driving motivation very often comes from the crystallization of both the backbone segments and the pendant segments. So I suspect that if we don't have experiments, but PEG and hydrocarbon are predisposed to microphase separate. In the other case we have done is put aromatic units on one segment and no aromatic units on the other. There is no amphiphilicity, but there is a rigidity contrast, which also gives you some value for the Flory-Huggins parameter. But whether there's a lower limit for allowing phase separation, I actually don't know the answer to that, Binny, but there would be. I think if I have hydrocarbon in both segments, it will still fold, Binny, because there have been experiments done on polyethylene where you have periodically located substituents, and just the bulk of the substituent allows the system to fold. So it's not really a Flory-Huggins parameter driven, but just driven by the bulk of the substituent that cannot pack in the lattice. So it's a little, I mean, the answer is that yes and no. In some systems, the Flory-Huggins parameter will not play any role, but in other systems, it will have some effect.
Thank you.
Professor Vasudevan will take over now.
Okay, thank you, Professor Jemis, and thanks to Debashish, Tilagar, and Arunan for giving me this opportunity to participate in this meeting to felicitate Ashok. So let me now invite the next speaker, Dr. Shrabani Dinda, who was formerly a student with Ashok and currently at Schrodinger Incorporated in Hyderabad, to share her talk on catalyst design using molecular modeling and data-driven approach. Shrabani.
I will share my slide first. Okay. Is it visible? Yes.
It is.
Okay, I am audible, sir?
Yes.
Okay, okay. Thank you, Professor Basidevan. Thank you, Professor Tilagar, for giving me the opportunity. And thank you, Professor James, who has always been inspiration to me, and my other Professor Arunan, Professor KLS. I love because my scientific journey started in this lab, and we are gathered here for Professor AGS. And this is a photo, I think one of the IAC day, then Durai Pandit just joined, and they are my seniors, Rakesh, Akshay, AJS, Durai, Ramchandran, Jabudurai, and Sangeeta. And this is a photo when I bought my first camera with my scholarship, and I came to sir's office then and asked his permission, and he was so happy to give the snap. So thank you, sir, dear sir, thank you so much. I'm always blessed to have you in my life, not only as a PhD supervisor, but you are a fantastic human being, and I learned many things in my life, not to become a researcher, but to become a better human beings. And my journey started in this lab was not so easy, because I finished with completely computational work, and it won't be possible without you and the supports I received from entire department, especially Professor James, KLS, Arunans, and their students like Usha, Mosami, Parameshwar, Shushmita, and others. Thank you all. So yeah, now I will start with my current research, and some of the research I have started after I joined Schrodinger. So we so far we have all seen mainly the experimental work, and to certain extent some of the modeling work to understand what's happening inside, which we can't see but we can understand. So the data science is sometimes coined as the fourth paradigm of science, because this century and with the coming times, we are flooded with the data, and it's coming more and more that if we know how to organize the data, if we learn a lot from data. And it's been very much visible in other sectors like advertisement, and in biology, image processing, traffic controlling, and so much. And it's now become very much encouraging for chemistry. Other than doing the molecular modeling, we can extend this approach to the data science and learn new things, which in turn provide new insights to make a better decision. So we can find so this data is a data not only from we generate the theory, but it's a data including everything, including experiment, including research. And the challenge is to gather the data and to analyze the data, and then to find a pattern in the data, which in terms help us to to get into more about particular reaction or particular scenario. So here, since I have been working with people, AGS, JKB, and later Notka Roche, or other group recently with Professor Sastry. So there I've been very much with the catalyst. So I thought how this catalyst design we can do with the data science. So before that, let me show you some of my work which I did for molecular modeling perspective. So as we all know that it's all started with the real system in experiment, and then we need always a model. And then some of case we have seen with the model system, and sometimes if model is how good the model is, that is the thing. And then we just do calculate several steps, elementary steps, and our goal is to find the rate determining state and concentrate on some of the crucial intermediates which governs the reactions. And this is not only just the intermediates, it's driven by the additive solvents and supports so many things. And this in term, it's not only understanding the experiment, but it's also telling the experiments from the modeling study that you can design the experiment or do this study in order to understand the reaction in a better way. And data-driven approach is just one step away. We are not doing any transition state calculation, or we are not doing any synthesis, but we are just gathering the data, and from there we are extracting the information. So when I started my journey in heterogeneous catalysis in Singapore with Professor Notka Roche group, so we had a very exciting problem with the rhodium embedded zeolite, that is a fugicide one. In one case, you get ethane exclusively, whereas in another case, you get butane as an major product. So as I mentioned in my earlier slide, this is a like zeolite, it's case system. So ideally we should do the periodic model, but here there are hundreds of possibilities how the molecules interact and undergo several elementary steps in order to get the final product. So what we did, we wanted to get some reasonable economic means computationally economic model. So we use the QMMM approach and giving the periodic as a standard, and then we analyze the data. And there are many elementary steps. This is just one of the pathways. As we can see, I'm not telling in details, but this is how the approach we generally adapt in order to do the catalyst modeling. So this this is a part of the zeolite, and this is a rhodium embedded. So the interesting thing which from the series of experiments from the Gates group, we saw that when you have ethylene, you exclusively get the ethane. But however, when you have ethylene, you just increase the amount of ethylene in the reaction mixture, you get butane more rather than ethane. So the question is like, what happens if you give, because you have a similar experimental setup, you just increase more ethylene flow and suddenly you get the butane more. And the problem is that whatever the paths we studied initially, all the paths lead to the ethane as the main product. So it was a very hard problem for us to get like why we are getting the butane. Then we thought that rhodium actually can accommodate more than 2 ethylene. So we giving the third ethylene. It was not that sterically crowded, rather it was stable. And then we explored the different CC coupling pathways, and one of the pathways leads to the butane. So from this experiments, what we understand, experiments, I mean the computational modeling experiments, what we understand that we could rationalize the butane formation, but not exactly the ratio what we were expecting, because both the pathways are very similar energies. So this is one of the approaches generally we do. Most of this calculation been done with DFT QMMM, and the reference was the periodic one. So this is another problem which I was working. This result is not published. So here is with the zeolite, but it's with the ZSM5. It's methane to methanol transformation, and inside you have the oxidizing agent hydrogen peroxide and water, and you have iron. So it is a very, very nasty system. However, you get, this is the first time the from Hatching group, they showed that the conversion is very high, and that was room temperature with water. So it's a very exciting thing. So I thought that the water and hydrogen peroxides are there, so there must be a lot of dinuclear system that may operate. So I just I will just open. Yeah. So this is, I summarize the results. I'm not telling in details, but just the way we generally approach or address the issues. And then there are, I try to find out what could be the possible dinuclear systems, whether it will be hydroxo or oxo, and then you have a hydrogen peroxide. So you have superoxo and hydroperoxo, those kind of possibilities. And then one case, if you just use just only one center, or it's like a dinuclear assisted center, something like that. So this also included a lot of intermediates, and it's a very complicated network. But finally, I could find the rate determining state, and it shows that bifunctional mechanism was operational. So this is another approach generally the computational people do in order to comprehend the experimental thing. Now I will show another example where we are not showing the whole energy profile, but we are interested in particular steps that we call the rate determining step, and that is the step which determine the enantiomeric excess in for a particular reaction. And my journey in this area started in AGS lab only. This was done by Rakesh, and then we observed that there is a very interesting like if you change this R1 and R2, you get a different results. I'm not showing in the results, just the approach. And this CC coupling is the rate determining step. So the thing is that you can see in that rate determining transition state, if you have the substrate and that catalytic backbone substituents, there are so many rotatable bonds. As a result, you will get a lot of conformers. So we just initially with the molecular mechanics scan the conformers, and then it's a systematic way from MM2 we did a QMM transition state, and then we use a energy cutoff of around 20 kilojoule, and then we take the final few structures around 10 to 15 for the QM calculation, and then with that full QM calculation, using the Boltzmann statistics, we calculated the enantiomeric excess, and with all the 5 catalysts, we got excellent experiment and the theory alignment. And then because I grown up in experimental lab, so when I came to IICT, I got several calls from the experimentalists like Dr. Rajesh, later on Chandrasekhar and Rambabu. So they have these are all organic catalysts, and this is one such example. And the transition state involved multiple hydrogen bonding, so I had to struggle a lot to find out which will be the most reliable or stable intermediates. And as we know that it's an enantiomeric excess, so we have both R phase and C phase analysis, and finally with the Boltzmann distribution we came to the conclusion whether R or S will be the major product. Well, now I will move to slowly data science. This field I've been introduced by Dr. Shastri when I joined in IICT. In their group, they were designing one web portal called Molecular Property Diagnostic Suite, MPDS. It was initially designed for disease, for a particular disease, so we called it a disease-specific web portal, and it has it's embedded. There are several chemoinformatics, CAD tools, and other computational platform techniques that is on a single platform. So I'll show in a moment, in a very brief, how it works. In MPDS architecture, so first thing is very important, we need to know the data. And data, believe me, though there are a lot of data, but it's not always easy to get the data. So first thing is that you have to get the data, and then you have to process the data. So that takes a lot of time. So in data science, most of the time goes for getting the data and processing the data. And when I say the data library in MPDS, it's the one portal is for the disease specific web portal, and then another one is with for the compound library. I'll show brief what it is, and then another portal for fragment library, and there is the another one, the catalyst library, which I was working. I'm still associated with it. And once you have the data, then you have to process it so you can analyze it. And for the analysis, there is something called the descriptors that is associated with the molecular properties. It could be like QM descriptors, or it could be your topological descriptors, or it could be constitutional descriptors, like that. That actually help us to map that, or rather than I should say that these descriptors correlates with the activities or properties of that particular molecule. And this advanced module, we are not yet there, but of course most of advanced multi-scale modeling, you can do it for the data analysis. And till 2017 and 18, they have around 124 million compounds, and those compounds are divided in several classes and clusters based on their architectures. And one interesting thing in MPDS, something is called the MPDS Aadhaar card, that is very unique. So you have a core of card number for the molecule, and that belongs to a class and clusters, and its serial number. Once you click it, you get also several properties like that, and these are the properties which I'm calling the descriptors. It can show you whether it's a drug likes, or it can show you whether it's a catalyst like. And this is around, I think, 23 publicly available databases. So the data are collected from there, and this is the current compound library architecture. It's still under progress. One of Sasti student is working on it. So initially it was designed to define them or distribute them in terms of class or clusters, but later on they thought it's a better way to divide them in layers, and then each layers in different portions. So this work is also not yet published. Okay, so coming back to the catalyst design, the underlying principles is very similar. So first thing, we have to get the library. Here the library is not for disease or not for anything, but it's for the catalyst, and it could be anything, literature, patent, thesis, or if it is from industry data sets. And then is the processing, and then we use a different machine learning algorithms to establish the quantitative structure catalytic activity relationship. And in order to do that, there sometimes the features you have to calculate, or you have to engineer the features so that you get the best model here. So this is a flow chart I already mentioned. So here in the data classification, we have currently I'm working on this homogeneous organometallics, and there are few types of reaction which we are interested in. And we have a database for metathesis graphs, so Hobeida-Kai metathesis reaction, and some of for ethylene polymerization with Princer kind of catalyst. So, and once you have the data, then you organize the data, you calculate the descriptors. There are several tools available, many are free, and then you establish, then you do the data analysis and establish the model using machine learning method, and then you predict that what are the these are the predictive modeling approach, what are the features or descriptors that makes your catalyst a better catalyst, and that in terms will help you to design a more efficient one. Or we can design in silico also and tell that, okay, you do functionality here and there, and then according to this model this catalyst should work. And in this direction, so currently I'm working in this ethylene polymerization reaction with this pincer kind of, these are the architecture. So I'm not telling you exactly the structures, because I'm not allowed to. And yeah, currently we have around data set of 266 and features of 584 descriptors, which we calculated with Schrodinger and some of them from freely available tools. And the mapping parameters, that is the activity. This activity is the experimentally known activity, and our goal is to find an automatic workflow so it will able to predict the catalyst or the features which one should keep in mind when they design the new catalyst. This is another example with metathesis catalyst, mainly all are the ruthenium. And this is not specific to a particular reaction, but that's all the metathesis catalysts, and I have a data set of around 870. Those are all experimentally known, but this is not for a particular reaction. So here, unlike the previous one where we want to show the activity, but map the activity, but here we wanted to get a pattern in all these 870 catalyst, and then wanted to map it like which are the particular patterns correlates to their activity. So here activity, though I'm not calculating and not taking any experimental one, but we are calculating from the barrier calculation. So for all these 870 molecules, I calculated the barrier, and that barrier is roughly proportional to the activity according to these references. So this result is also under progress. And generally, my final goal is to see something like that. So like I showed you, I have the features around 800, like for the pincer catalyst around 500. So from the 500 features, I want to see which are the best 30 features that govern that catalyst as the effective catalyst. And so these are the very important things. I'm not showing what are those things because
I think the time is running out.
Okay, yeah, this is my last but one slide. And we are sometimes having a model like that when we do the training set and test set, but you can see this is not that great model. We are expecting something like that. So this is, we use a different machine learning tool to get some model like this, and from there we extract the features like that. And in Schrodinger's, these are the modules we have, like AutoQSR, DeepChem is freely available. You can create the fingerprints from Jaguar or Canvas. And most of my work I did with the Python script that is available in public domain. And for the biological work, you can do the active learning glides also to get the docking dataset using BillionUp. I think these are some representative publication from Schrodinger's. And with that, I just like to thank the organizer once again, and Professor Samuelson, Madam, Professor KLS, Jemmy, Sarunan, JKB, who is here because I spent very qualitative time under his guidance in IIT Kanpur, and Professor Ross, Professor Shastri. Without his encouragement, I won't be able to get in this data science work. And they are my collaborator in IICT, Dr. Madhvi Shastri, Tanvi, they are my collaborator in Schrodinger. These are the funding agency. And thank you all for your kind attention.
Thank you, Shabani. I think we have time for one quick question.
Okay.
Okay, I think there are no questions, so thank you very much.
Okay. Thank you, sir.
Yes, well, there's a question.
Shall I stop sharing it?
Okay, I think you can stop. And
hello. Hello. Hello, am I audible?
Yeah, go ahead.
Yes, sir. Thanks for the opportunity. My question is regarding any data set is available regarding covalent organic framework. We have seen many of the other ligands, catalysts, and even biologicals also. the new trends like a covalent organic framework or metal organic frameworks? Any data set is available in computational?
Say once again, data set of which?
Data set of covalent organic frameworks, which is a new trends in.
Okay. That I don't know, but I don't think it is available because I have searched it, but currently I'm not sure whether it's there or not.
Okay. Thank you, Shabani.
Thank you.
So may I now invite Professor Pushpendra to share his talk on story of heresy. Pushpendra.
Das, your mic is in mute. Or
Das, your mic is in mute.
Yeah, can you hear?
Yes, we can hear now. We can hear.
Okay. You can see the slide. Yes. So I guess my talk time is already over, but I still would like to thank all the people who have actually put hard work to actually enable this meeting and taken, a lot of sort of time from their schedule to pay our respect to our dear colleague, Professor Samuelson. And in this, what I wanted to say is that when Tilagar asked me for giving this talk, I readily jumped into it because this is a talk that I have not given ever, and I'm not sure if Professor Samuelson has ever given this talk. And what I wrote in the abstract, the story of ferrocene and I put 3 dots, which actually disappeared when it appeared in the actual program. Okay, I'll tell you why I put those 3 dots, because I thought that what could be an appropriate title that I'm going to actually say for my colleague. So I thought of the next title was The Story of Ferrocene and My Lifelong Collaboration with Ashok. This is a Bengali way of saying his name. He is actually Emperor the Great, and his name is spelled exactly the same way, which actually it means, and he's really great. But then this is my way of actually spelling his name, and I have been criticized by many people for this. Then I thought, okay, the story of ferrocene and my research journey with Professor Samuelson. Okay, and then then there is a mundane title I could have written, The Story of Ferrocene and Its Influence on the Inelo Properties of Molecules. Now you can actually take whichever you like, but that's why I actually kept 3 dots, because all these 3 titles I'll defend by the end of my lecture. What we decided to do long time ago was look at second harmonic light scattering from molecules which are actually dissolved in solution in a particular solvent. Now, this technique was initially invented by the French physicist Terhune and Maker, and it was initially used for looking at structures of liquids. Then later on, in about 1991, then Parsons and Clays, they used this technique and revived it for finding out second-order nonlinear optical properties of molecules. Once this actually hit the journal, then a lot of people got interested in it, and we also got interested in this technique. What it basically says is that if you actually shine light which has a dynamic electric field associated with it, you polarize the molecules, and in that polarization, you have the first order polarization, you have the second order polarization, you have the third order polarization. So in electric field, you can actually have multipolar or multiple order polarizations, and you can actually measure each of these tensor quantities, which gives you an idea about the polarizability of the molecule in that order. So we decided to work on the second order polarizability, or the first hyperpolarizability of molecules, which is characterized by this tensor beta, which is the third rank tensor. We'll not go into the details. So what we did actually, we we had some laser in our lab and we were doing some other work with the laser, but we just put together this experimental setup. In fact, in those days, there was a very able graduate student named Paresh Ray. He, in fact, actually established this technique in my lab. And in this technique, what you do is you shine light on a sample, which is actually kept here. And then the second harmonic light that is generated, which is twice the frequency and half the wavelength. So if we shine, let's say 1064 nanometer light, which is normally what we use, we get 1064 nanometer light scattered. Of course, that's the normal Rayleigh scattering, but also we get 532 nanometer light scattered, which is the second harmonic Rayleigh scattering. And that light is collected through a monochromator and then, of course, processed. So, but intensity of that light is related to the second order polarizability, which is beta, as you can see in this particular slide, where this beta solute is the second order polarizability of a molecule that you are going to measure. And this expression has a linear relationship with respect to the number of solutes that you use in solution. And the solvent contribution is there, but solvent contribution can be actually taken off by using a reference, which generally is paranitroalanine for some of these molecules that you have measured. And with respect to paranitroalanine, if you know the slope of the straight line, as it is shown in this figure, with respect to the concentration or number density, then you can compare and you can actually find out the beta or the unknown polarizability, second order polarizability of an unknown molecule. So that's what we basically do. And that's what we decided to do at that time. So we actually were wondering, both me and Professor Samuelson, in terms of what kind of molecules that one would try. So we decided to try ferrocene. Now, why ferrocene? Because ferrocene is an interesting organometallic molecule, but it has two handles, the cyclopentadiene ring and the iron center. And this can be easily integrated or grafted in any structure, either through the cyclopentadiene ring or by coordinating to the iron center. So normally ferrocene is a donor, but this donor status of ferrocene can be changed by if you actually just oxidize the iron center. So that is why we wanted to see how integrating ferrocene in a molecule or a part of a molecule or is part of a large structure can change or alter the second order polarizability of the entire molecule. Now, there are two reasons that why we didn't want to actually use it. And one is that it has a strong absorption in the visible. Now, this absorption is loosely can be actually said due to the DD transition, as you can see on the right, there is in the visible, there is a huge, large absorption band. And this absorption is detrimental to our measurement because it generally interferes with the second harmonic signal. And one could, of course, make in principle correction. But what is much more difficult to make is that not only the single photon fluorescence will be there, but there will be also two photon fluorescence, because at 1064 nanometer, if you use two photon, you can excite the fluorescence in this part of the absorption spectrum. And that correction is a lot more difficult to do. And that will artificially increase, as we will see as we go along, the second order polarizability or beta of a molecule. Now, also, ferrocene has a high degree of symmetry. So ferrocene as such has a very low even order polarizability. The reason is that it has a D5H symmetry, but actually D5D in solution because these two cyclopentadiene rings actually rotate. And all even order polarization tensors cannot be sustained with this symmetry. I'll not go into the details of why, but so therefore ferrocene itself has a very low contribution to beta. But let's see what we can do with ferrocene. And at that time, so there is this paper where actually Seth Marder and Green, they made this molecule, which is, you can see that is a simple molecule which has a nitroaromatic ring on the on side and the ferrocene. And the ferrocene was actually the donor, and the other ring, the organic ring, was the acceptor. So in this donor acceptor molecule, they found that in the powder form, this molecule actually has a very high second harmonic efficiency, and it's 67 times of urea. Urea is an organic molecule which was thought of to replace all the inorganic molecules like KDP, ADP, the potassium dihydrogen phosphate, the ammonium dihydrogen phosphate, and the beta barium borate in those days, because the organic chemists were trying very hard to come up with second harmonic generators using organic molecules. But then what happened, let's see, then we first make these molecules. Now, Professor Samuelson will actually tell you how these were made. There's a student of his named Allegation. He was actually here a few minutes ago, and he's the one who actually made all these molecules. Now, these ferrocene molecules, when you actually couple with DDQ or TCNQ or iodine, then you can actually form a strong charge transfer complex or redox complex, which actually will have large beta values. And for example, if you combine one with this TCNQ, which is the D here, then you can get a beta value of, let's say, 6 in this unit. Now, this unit later on, I'll not actually show every time, but it's in beta is always mentioned in this 10 to the minus 30 ACU unit. And then, for example, the 2D has 132 and 3D has 246. So as you increase these lengths of these molecules, and you also use the shift-based linkage to couple this organic, I would say that this is kind of a donor-acceptor-donor kind of a structure, or a donor-acceptor structure. But these are all neutral molecules which you can basically oxidize by using the strong oxidants like DDQ, TCNQ. We'll see that later on we have used chloranil, TCNE, and so on. Now, there's one good reason that I have put, not by mistake, that is written TCNQ2. So what happens is, when you actually transfer one electron from the ferrocinyl compound to the TCNQ or DDQ, the resultant radical anion also associates itself with a neutral species. And that's known because the crystal structure of that species was apparently solved according to Professor Samuelson at the time. And he will answer all the questions regarding this. So we always wrote this. And then it turns out that if we actually just simply take the delta E half of DDQ, let's say, and one of the ferrocene compounds, then we actually can see a linear relationship of beta with that. And so we were a little bit excited. But the problem was that we couldn't characterize some of these complexes very well because of this radical anion combining with the neutral and so on. So, Allegation and Paresh published this paper. But around that time, Professor Jean-Marie Lane was actually making these donor acceptor compounds, where he actually integrated ferrocene as a donor in a carotenoid system. And they were measuring the mu beta by E-Fish technique, which is another technique. But they also found similar correlations and so on. And the student whose thesis that actually appeared, this work appeared, Mireille Blanchard-Des, actually she later on became a collaborator of mine. But this all started with these stories of looking at ferrocinyl compounds. Then, by that time, Allegation had to graduate, so he left. And then Susanto came into Professor Samuelson's group. And Susanto actually made a large number of molecules, again, with similar features, but a little bit of variations were there. And then, by that time, we realized, as I said earlier, that the ferrocene has this absorption band, and those bands, which is actually the metal to ligand charge transfer bands, you can loosely call them. Although, if you look at the molecular orbital diagram of ferrocene, you'll see it's a lot more complicated, and all the ligand orbitals and the metal orbitals are all overlapping. So, but roughly speaking, we always say that the ferrocene is colored because of the DD transition is an iron and so on. And these are all ways of simplifying it. But having kept that in mind, if you see that some of these molecules have a beta value, but by that time, now we could actually correct for the beta, and we can actually find out the static component of beta, which is beta 0. which basically takes care of a apparent correction due to these resonances at 1 photon or resonances at 2 photon. And therefore, beta by beta 0 in a two-state model can be calculated by using this simple formula which is written here. So if you know, therefore, beta, which you get from the measurement directly using that equation, then you can get beta 0. And you can see that, that if you take the TCNE complex with all these 4 different molecules with R as hydrogen, then you get the beta, which is actually very high. You get beta values of about 240. But when you actually calculate beta 0, which is shown in yellow highlights here, you get about 20, 40, something like that. So it's not very high. Intrinsically, the beta values of these molecules are not very high. So that was sort of a little bit disappointment. And at the time, Sushant and Anu, who actually did the work, Anu in my lab, Anu Krishnan, and Sushanto Pal in Professor Samuelson's lab. So we published this paper, and we published it in Journal of Organometallic Chemistry. And the reason is that we still didn't understand that how to correct and experimentally for these beta values, and the beta values were not very high. At that time, I remember Professor Samuelson was a little bit unhappy because we actually wrote this paper. There are a lot of interesting chemistry and insights that are given. And recently, when I was preparing this slide, I actually checked, and this paper has about 100 citations, including 2 in this year, in 2021. So if a paper like this have survived 20 years, I think Professor Samuelson has done a very good job in making them and studying them. So I'll not say anything more about citation because I know that many of you probably have your own opinion about this. So, oops, I'm moving. Then Sushanto made some more molecules, again in a similar fashion with ferrocene. And then we are comparing what will happen in case instead of ferrocene if we have a benzene ring. Okay, and these are all again symmetric structures, and you can see that they have large beta values, but then when you calculate the beta zeros, they're about 10 times as low. So that means, in fact, the actual intrinsic polarizability, hyperpolarizability of these molecules are low. But because of 1 and 2 photon contributions or resonances, it was artificially jacked up. But then by that time, we have another postdoc who actually joined. He's a physicist from IIT Madras, Nanda Kumar. And Nanda Kumar actually said that let's do this experiment where we can actually scan the second harmonic light through a monochromator. And we didn't, of course, have money to buy the monochromator. So we actually retrieved one from one old applied photophysics lifetime instrument that Professor V. Ramurthy left in organic chemistry department. And I actually took a monochromator, which was not a great monochromator, but you can see that the second harmonic light, which is at 532 here, and then you see it's actually followed by a broad 2 humps fluorescence. And then we actually fitted these to the second harmonic signal was fitted to a Lorentzian, and the other one to Gaussians, and then we figured out that we could actually extract the data, and then we wrote that paper in chemical physics. So this was about 2001. Now what happened, by writing these 3 papers actually, Professor Samuelson became very famous in the field of ferrocene chemistry. The Journal of Organometallic Chemistry was coming up with a special issue, and in fact, Professor R.W. Adams of University of South Carolina, he was actually putting together a special issue in Journal of Organometallic Chemistry celebrating 50 years of ferrocene. So he actually wrote a letter to Professor Samuelson to contribute to that special issue. And by then, as far as I know, he only published not many more papers on copper than iron, and these 3 are only in ferrocene. But anyway, so then Ashok and I had to think about what can we do new to write in this particular Journal of Organometallic Chemistry. So at that time, actually, I was looking at supramolecular complex formation between melamine and cyanuric acid. Now these 2 molecules, they form complexes of different types depending on the substitution that you have. So we were actually asking that can we get two things from this, because two things that we always wanted to know. One was the stoichiometry of two different molecules which come together and form supramolecular complex, because the stoichiometries in solution is very difficult to get, particularly if it is not 1:1. 1:1 you can get easily by NMR titration and so on. But if it is 1:4 or 2:5, they're all difficult to get because you cannot crystallize them. So our question was that can we get them? So to start with, we started with these two molecules. By that time, when we started, Professor Whiteside must have written 100 papers on this particular couple, with of course different variations and so on. So he told us that you can have linear tape structures or crinkle tape structures or rosette structures. Now we took the simplest one, the rosette structure, and the first question that we asked that if we actually titrate, okay, by second harmonic, by adding one into the other. So what we did is we actually go back. Do I go back? Okay. So if you actually take, let's say, melamine in some concentration and you titrate it with cyanuric acid and look at the second harmonic intensity, what happens is you can see this solid curve, so that the intensity keeps increasing as the hydrogen bonded complex gets formed. And once you hit 1:1 ratio, it becomes more or less flat. It goes up a little bit, which you cannot see in here. And then I took that compound from Professor Ramakrishnan's lab. He was generous, and where you have actually blocked these hydrogen bonding sites by putting phenyl. And so that other one is the one at the bottom actually is that control experiment with the triphenyl. You don't form that hydrogen bonded complex. And these complexes are formed in chloroform, but if you add methanol at 25% volume, you can reverse this and you can actually dissociate this hydrogen bonded complex. So now Professor Samuelson made these 4 wonderful molecules, 1, 2, 3, 4, and we wanted to actually see that here, of course, in 4, you can see that the hydrogen bonding sites have been actually blocked by putting methyl. And so this is the control molecule, and see between these what kind of complexes will form and whether we can identify the stoichiometry of these complexes. And so now in the board, you can always justify any structure. I can justify 1:2 complex and 1:5 complex between 1 and 3. Okay, and they're perfectly defensible. I'm not cheating anywhere in this. Okay, now the question is that now we did the experiment. Again, the second harmonic titration, adding one into the other in chloroform, and we found that actually between 1 and 3, okay, you form actually the 1:5 complex and not 1:2 complex. And actually, but if you have the control experiment, then actually it kept going. But at some point, what happened is that it was coming down a little bit. Now we never actually knew why it actually came down at that point, but it is reversible. Again, you can add methanol and you can reverse it. So finally, we submitted this in that 50 years of ferrocene issue, and that was accepted. So then we are actually thinking about, what you can do with using ferrocene. Okay. Now, there's another one which actually Millie worked in our group. Here is again, you can form a DAD hydrogen bonded complex with one with the other. Now, this molecule, if you actually freeze in this particular configuration, then it's a lambda-shaped molecule. But in solution, there is no guarantee that it will be like this. It will be flexible. But then, if you bring another opposite molecule, oppositely bonding molecules, which is ADA motif, okay, then you can actually log these two in that particular lambda configuration. And when I say the donor acceptor donor, here they are hydrogen bond donor and hydrogen bond acceptor. Okay? It's not the electron donor and electron acceptor. But you can therefore make motifs of this type because we knew this. And so what we did again, we did a second harmonic titration, and we found out that you actually have 1:1 complex of these two. And beyond that, nothing happens. And then you can again dissociate it because that's a test you have to do. And we also did this, in this particular case, it was 1:1, so we did this by NMR titration. because one of those NH proton chemical shift, okay, was changing as you added the other one. And it exactly follows similar pattern like the second harmonic. And the Job plot is here. And in the Job plot, you can see that you form a perfect 1:1 complex. So this was simple. What was then we were actually looking in this particular molecule? What we are looking at, that if you see the dipole, the direction of dipoles, once you engage the ferrocenyl moiety with the amide, organic amide molecule, then what happens is that you reinforce the dipole. And the beta of individual, like the organic moiety, the beta is 45 in the same unit, remember. This beta is 55 for the other one, the ferrocenyl moiety. But when you put this 1:1, you don't get 100, but you get 550. So you actually enhance the second order nonlinearity by forcing this and locking this into a lambda-shaped configuration. And that is because that your actual dipole moment becomes huge when they actually come together. And it becomes unidirectional. So this was done by Mealy, because Mealy also knew how to synthesize molecules. So Mealy did this, and so he published this paper. Then, by that time, we did some other things, which I'm not going to discuss because I will keep it into ferrocene. And so by this actually brought us to around about 2016, when Karthika came around. And Karthika formed these ferrocene moieties, where actually you have coupled a thiophene with ferrocene. And now thiophene with ferrocene can be coupled. You can have two ferrocenes actually substituted here, or all the four hydrogen can be replaced or substituted by ferrocene here. And of course, these synthesis procedures are known, and Professor Samuelson actually instructed his student, Lakshay and Karthika, both of them, to prepare this and of course characterize this. What is interesting about these two molecules, that now, by now we had actually a lot of facilities. One thing is that we could now, instead of using chemical oxidant, where the reduction was very difficult, we can now do electrochemically. And then we actually made this cell by this time. And so here you can do in situ electrochemical oxidation reduction by using electrodes. So we have set up a potentiostat inside the measurement cell. We can actually look at the absorption of these compounds as you actually oxidize or reduce them. And that is through a Avaspec in situ spectrometer. At the same time, you can make second harmonic measurements. So these facilities were not there in our lab when we did the first set of actually experiments with ferrocene. So we had to use chemical methods of oxidation, but we couldn't reduce it. But now we can actually, in these molecules, we can nicely, one by one, we can reduce the iron centers. And you can see the CV is very clean. And believe me that it actually is about 1 electron transfer in each state. And you can reverse it nicely. Okay, it's very difficult to generally find such nice CV patterns, okay, in electrochemistry. And I was actually impressed myself that we could find and actually identify all of them. And then here is the experiment, and here are the potentials at which all this oxidation and reduction of compound 1 and compound 2. Remember, compound 1 is a bisferrocenyl compound, the other one is tetraferrocenyl compound. Okay, and it's on thiophene. Now it turns out that for the neutral compound of the tetraferrocenyl thiophene, the crystal structure has been solved. And it turns out that the cyclopentadiene and the thiophene, all the pi electrons are not in the same plane. There's a twist. And because of that, what happens is that you don't see very high beta value. And here are the beta values which are highlighted for 1. So if you do 1 electron oxidation, it increases from 25 to 55. So now what is happening is one ferrocene is Fe3 plus notionally, and the other one is Fe2 plus. So one is a donor and the other one is an acceptor. So you have actually created a donor acceptor motif through the thiophene ring, and that actually increases the beta value. The moment you actually oxidize the other one, so that means both are now Fe3 plus, again you are bringing in a symmetric thing and it goes down. It becomes 45.
Pushpendu.
So that hypothesis we wanted to extend to the 4 center. Now it turns out that it's not very easy, and the reason is simple, because if you see all the even oxidized species like 2+ and 2,3+, they have higher beta values than all they have higher beta values than all the even ones. So 2 to 2+ and 2 to 4+ have very small beta values. And what is happening, we believe at this stage, that they actually have a kind of a symmetry around thiophene that is developing, and that symmetry is actually not good or conducive for high beta values. Whereas for 2+ and 2,3+, there is 1 iron 3+ or 3 iron 3+. So there is a lot of asymmetry in the charges within the molecule, and that creates a little larger beta value. But we didn't actually see the kind of beta values that we wanted to see. So this particular experiment has been done by Karthika, and I have just sent a final draft of this paper to Professor Samuelson. So I think that we started in the 90s, and we are going to probably both retire around the same time writing that last paper, and that will be actually given for a Fedstrift issue for a good friend of both of us, that Professor Palaniandavar, which is coming up in 2 months. So we'll write this paper. So now you see that why I called it as a lifetime collaboration, because it's a collaboration that lasted for more than 30 years. And lasted around this theme, and there's another theme which I didn't talk about, where we were actually exploring the halogen effect on the nonlinear optical properties of organometallic complexes. Now, halogen atom effect is also important, but not as important as the ferrocene, as I have demonstrated. And the people who worked actually from our group, I've only listed them because we had larger groups. Paresh and Allegation, they started the work. Anu and Susanto made us famous with ferrocene compounds. Nandakumar, Mili, Karthika, Lakshay. Now Lakshay, Susanto, Allegation, and Raja, they are from Professor Samuelson's group. Somi, Karthika, Mili, Nandakumar, Anu, and Paresh, they are from my group. And of course, there are other postdocs who worked on this problem, but that were not related to this. Dr. Wahab, Brito Das, Ravi Kumar Reddy, and S.B. Chavan. Now, here is a picture that I found in old days, but unfortunately, Professor Samuelson is not there in the picture, but you can see Anu, you can see Mili, and you can see me, and you can see the laser lab. That was the kind of lab that we used to use in those days. And the apparatus that you see actually is a photodissociation apparatus, which was there in those days. And it was a great pleasure, Professor Samuelson, to work with you on these problems. And we have actually developed certain themes over the years. And IPC department, which is on the right side, and Indian Institute of Science, a great place for collaboration. And I have enjoyed my time with Professor Samuelson. I worked with a research supervisor who was one of the finest gentlemen in the US chemistry. And I have now collaborated with someone who is the finest gentleman in Indian chemistry. Thank you very much.
Thank you, Pushpendu. Thank you, Pushpendu. I think we have time for one question.
Das, just a quick question.
He's saying something.
So the porosity of the pressure is the square of does it go with the square of dipole moment? Because when you went from 100 to 550, you have been muted, I think.
Yeah, dipole moment, we couldn't measure. Okay, so that's the problem.
That's okay.
You need to measure. If you calculate dipole moment in such cases, you will not get the correct answer. That's the problem.
That's okay. We'll talk about it later.
Yeah, audio is somehow going as square.
Okay, I think we're sort of running out of time, so I think all questions can be deferred to a later time or occasion. So thank you, Pushpendu, for a very interesting talk.
Okay, thank you, Professor Gemmis and Gemmis and Vasudevan for chairing the session. Let's close this session now, and I would like all of you to join in the felicitation meeting, which is in another link, unfortunately. We'll see you soon there. Thank you.